Usually, it takes about 7 to 10 years to go from a discovery phase to clinical trials. That's literally usually the time frame we're looking at. In this case, I think that we could easily shorten it by half.
That was Elizabeth Pavez Lorie, CEO of CompreVie, a young biotech company focused on building highly accurate human organ models in the lab to improve drug and product testing. CompreVie's mission is to close the gap between lab testing and clinical trials by replacing animal models with human-relevant tissue models that better predict real-world outcomes. CompreVie is reshaping preclinical testing by providing platforms that embrace the reality of our body and environment. Welcome to the Biotech Stack, where we explore the cutting-edge technology fueling the future of life sciences and biotech. From next-generation lab automation and AI-driven drug discovery to breakthrough biomanufacturing and research tools. We dive deep into the innovations transforming health, food, energy, and the businesses behind them. I'm Corinne Regan, your host. And in today's episode, I'm joined by Elizabeth, CEO of CompreVie. Hear how Elizabeth's motivation stemmed from a lifelong interest in human biology and a personal drive to improve disease treatment shaped by having a father with chronic condition. She's trained in Sweden and Germany and now operates from Vienna, where she's co-founded CompreVie with CTO efficacy. Listen as we chat about CompreVie's next-generation organ-on-a-chip technology for human-accurate safety and efficacy prediction. Let's get started. Elizabeth, thank you so much for joining us today.
Thank you.
I'd like to kick things off. Can you walk us through your background and what led you to start CompreVie?
Mhm. Well, thank you so much for this opportunity. So, my name is Elizabeth Pavez Lorie, and I'm the co-founder and CEO, CSO of a company, quite newly started company called CompreVie. Well, my journey started many, many years ago, actually as one of my key interests in life, and that's actually to understand how our human body works and how we can solve problems to make diseases go away. This started very early on. I have a father that has a chronic condition, so that was kind of, it has been one of my key things, key north stars, so to speak, in this journey to see how I could find ways to treat him. And also, I was extremely curious about the human body, so that kept me going. So it was quite obvious in a way, even though maybe not for 14-year-old Elizabeth, that I would be going into that part of my career. And I got a PhD in Sweden, so I come from Sweden. And then I did my postdoc period in Germany. And then after that, we decided with the family to move to Vienna, where in Austria, where I am at the moment. And that's where I met this incredible, young, talented lady called Ifigenia Petrohilio, who became my co-founder for this company. The idea of the company I had for many, many years, since I kind of started my postdoc period, and how to make sure that we have the most accurate models that we can possibly have of ourselves, humans, in the lab. That was the idea. Um, and that is the idea that literally CompreVie has. It's the task that we want to achieve, to have these incredible models of humans in the lab. And why that is because we are another species to rats, to mouse, to any other, I would say, laboratory animal that is used. And with that, we have also seen already the failure rate of drugs and the failure rate of products. It doesn't need to be drugs, it can be products that you use, household products that you use at home, both cosmetics, skincare, or other products that fail to actually keep, I would say, the irritation or side effects at bay. That's the reality of it. Not to mention that pharma has a failure rate of 90% from preclinical trials to clinical trials at the moment. And that's partly because of this, because of the fact that we have not been able so far to actually model ourselves more in the first stages. So with that said, it's a big challenge. It's an enormous challenge, but we think that what we have been able, or I have been able to develop during the years, we are going to be able to start in a good way. So that was kind of, I would say, a very wrap up journey of this. Otherwise, I'm a mother of two. I have a dog, a husband as well, and so on. And Iphigenia, as I tell you, she's this young, devoted tech engineer with the enormous, I would say, talent to actually be able to talk about technology to anyone, and that's a gift. So I'm very happy to have her as my partner. Mm.
Well, that's a great start to the story. You've alluded to it a bit, but
Mhm.
can you go into a little more detail on what CompreVie focuses on?
Yeah, our focus is to make accurate models of our organs, our tissues in the lab. So, not just cells that you then grow in the lab and then you put some substance there and then you see what happens to it, but we really want to be able to make models that are true, I would say, true to what we see in vivo in our cells. Okay. And with that, it means that it's a challenge not only to make sure that you have the correct cells in that model, but also that these cells are in an environment that represents them. Now, most of these, because there are already models that are doing a lot of these things already, quite good models that are doing that. Our model is different because, again, the viewpoint is to make it as accurate as possible. So, one of the challenges is, and I mean, was that we wanted to make sure that the environment that these cells had was accurate enough. And the only way to do that is to actually do what our body does, meaning that our cells themselves produce their own environment. We are not eating something or transplanting something that makes a tissue in a normal circumstances. Actually, everything that happens in our body, our generation of tissue, is something that is produced inside that cell that is then secreted, and then you start building up this incredible tissue and then an organ. And that is what we really focus on, that it's so genuine and so authentic that you can say, okay, these cells, we're giving them all the goodies and all the things that they want, and then they are able then to make their own tissue. They're able to make their own environment. This means that we are able to put them in a balance with everything else. And then what happens is that they are so happy, so to speak, they're so happy in where they are, that they can be kept, of course, in culture for a long time. Because that's the other challenge that we have at the moment in preclinical trials and in laboratory trials, okay, in vitro. And that's that we are at the moment only looking into acute, so the very beginning of what happens. So, for example, how toxic something is after 24 to 96 hours, okay, or what something happens within a window of a few weeks, 2 weeks or three. That's something that is super important. But the fact is that most side effects that are going to be a problem are those that come afterwards because of repeated administration of drugs or repeated cream applications or whatever you want to call it. So this is the other part, the part that is the chronic application, the chronic use of something. And that is not being able until our system to be done in the lab. So our models are be able to be kept in culture for 6 months. That means that you can actually do a lot of stuff and you can get a lot of data out of this, of course, because you can keep them there. And the idea of the system that we have is that we can collect also data without without taking out this sample and doing the analysis on that sample, and then that sample is gone. So that we can follow, like we follow, for example, if you would go to a clinical trial now, they will, of course, follow you through time. Okay, they will make some tests, but they will not destroy you in the process. It will be you giving some blood samples or you giving something, but you will go home, you will do your things, and then you will go back and they will take maybe more blood or something else. So that's what we want to achieve, but in the early stages with this really, really human-like models. So that's that. It's really the emphasis of making sure that whatever is happening in the clinical trials, that we can make that happen already as much as we can in the lab, in the laboratory. Okay. So we don't need to use other animals. So we don't need to have too much space, too much time going from this to this, that we can narrow the time space. And that due to this narrowing and not using other animals, we might be able also to reduce the costs. Okay. That would not be bad, so that we can actually make sure that we are able to close this a little bit more than what is now, because now it's a big gap. You have clinical trials here, and then you have what is happening in the lab in vitro here. It's really, really a long way to go from what you can get here to clinical trials. This is what we want to kind of make sure that they come together more. The name of the company is CompreVie. It means that it's literally a fusion of two words. It's a fusion of comprehending something, and in this case, comprehending life. And the reason for that is that we as humans are not static. We change with age, we eat different things, we go outside and we are exposed to other pollutants or non-pollutants, it doesn't matter, we're exposed to things, okay? None of these things are usually modeled either in a laboratory. So it's a very, I would say, sterile environment, because you want to, in this case, only include something that is, that you know, okay, if I put this substance there, I just want to know if what happens if I put this substance there. And usually you have already thought about an endpoint analysis. Okay? For example, upregulation of P53 or whatever you want to analyze. You have already thought about it. You have something. What we want to achieve is to literally embrace the fact that we are so chaotic and so heterogeneous and so enormously complicated, okay, as humans. And for that to happen, you of course need to have models that are complex, need to have also infusing in this a casing, which is the chip that is built into with this biology. And this chip will then be able then to collect the data that we, in this case, are getting from this biology inside it, okay? So collecting that data, which is, we hope to be able to get so many things from it as we possibly can, and then use this really cool technology that is making a lot of heads turn at the moment and is seen as an enormous step for humankind, which is artificial intelligence, but use that for prediction. So, because as you know, we may be super complicated people, super complicated species, but at the end of the day, we see things only in two dimensions. We are not capable of comprehending more than two dimensions at a time. So why not make sure that we have this incredible tool that we could collect all the data, and then we ask that tool, okay, so in this experiment, this substance X was put in, and what do you know about, I don't know, upregulation of tumor suppressing genes? Okay. And let's say that we have collected a lot of data on gene expression and so on and so forth. So then this AI concept, this predictive concept will tell us, yeah, this is what we know. And this is the prediction that I can give you for this type of individual for if this individual uses XYZ. Okay, this is what we would like kind of hopefully 10 years be in and so on. Now we're just starting. So the model is there, the MVP is there. We have been so lucky to have already pilot testers that have been able to use this very, I would say, prototype like concept. But we are happy that it has worked well and that it has served a purpose for those that have tested it so far. Now, the organ that we started with, because you need to pick one, you can't go like completely crazy and go for all of them because that will take you enormous time. That would be nice in academia, but for a business, you need to start thinking, okay, I still need to kind of have a deadline when to present this to the world and in this case, be a commercial entity. So, with that said, we started with the skin, the human skin, as the organ, but we have a purpose of course, continuing with other organs after that. So, it's not going to be only the skin, even though the skin is extremely important for a lot of different things. It's one of, I mean, it's the organ that we are mostly exposed to the outer environment. Not to mention that it is about 25% of all the, I would say, the tests that you do for any kind of situation, be it in pharma or in other concepts, will be in skin. So it is a very good tool to start with. I also spent, I would say, almost more than 15 years working on this concept. So this was also the I would say the good advantage that we had and why it was in a way so easy to go from launch in January 2023 when we launched the company and to have an MVP within 6 7 months. And that was because Iphigenia, with her enormous fantastic brain when it comes to technology, she already had a concept that she wanted to try when it came to the ship, the housing, and so on. And the biology was already developed for many years, so it was quite easy, put it like that, because it's never easy, but it's easier, I would say, easier to fuse it and then that to be able to test it and have a result. And we were lucky enough that the result was good enough for us to use it as an viable MVP that could then serve people, and we could get some feedback also on what they think um, that they would like to do.
To go into a little more detail, we've talked about timeline. In addition to the cruelty of going and doing animal testing,
Yeah.
the timeline itself, where
Mhm.
going from animal models versus going to something that is more like
Yeah.
what's going to happen in clinical trials. Do you know what that timeline currently is? And do you know what that timeline could become?
I would say it like this. Usually it takes about 7 to 10 years to go from something, a discovery phase, something to clinical trials. That's literally usually the time frame we're looking at. Now, we
Okay.
know for a fact, and we know this because we had this COVID, and there
Yeah.
we needed to be very fast. In this case, I think that we could easily shorten it by half. But it depends on having all of this kind of done in a regulatory concept that we will need to then think about more, because now regulatory is still quite, I would say, based on 2D concepts, on just making sure that you have cells there and you try something in an acute phase. There is no real kind of notion how to handle this with regards to chronic in vitro at the moment, things like that. So the regulatory space on that um, will need to change on how we think about this a bit more. Do I think that this is going to be something that is happening soon? Yes, I absolutely do. I think that more companies will be going into this direction. Maybe not how we are doing it with regards to having our own biology that we ourselves do and that we use that to predict or to do other stuff. But maybe they will do it in an in silico concept, meaning that they will use only database knowledge they acquire from publications or from already clinical trials, and they will do predictions on that. So all of those things, all of those other tools can also be used in this space. Okay, and the more of these things that you can use, and depending on, I would say, the how this company, this pharma company or other company wants to reduce it and wants to use it, you can of course think of different ways of doing that. But in my opinion, we could achieve half of the time. Yes.
That's incredible. And then
Yeah.
talking a little bit more about regulations,
Mhm.
it sounds like there's regulations that are starting to change around
Mhm.
animal testing in general.
Yeah.
But you've mentioned this a few times now that there's acute testing happening, but not a lot of chronic testing,
Mm.
and especially not in the same environment.
Mm.
And so you do also hear or believe that the regulations around that type of testing will change as well. So it's like a two-prong effect.
Yes, at the moment I think that we're still unfortunately kind of not struggling, but we need to kind of raise the awareness of the fact that there are models that could actually work for a larger space than just the first part. Okay, and that there are predictive tools like in silico concept that can help as well. All of these things, okay, raise the awareness of this even more and show really that this is possible, which has been around for a while now. But I think that it's changing because of the governmental, so top-down concept, where in this case governments say we're not going to let you use this anymore. Okay, and then they need to start thinking about a solution, and then of course they see that there are actually companies and institutions that actually have technologies that could help them. Okay, and this is the way that it will it will change even more. And I see this more accelerating more drastically now because you have a lot of this going on, top down concepts that are coming up. I mean, the FDA has a lot of that already. You have the European regulation that is looking in Brazil, India, Korea, Australia, I mean, Canada, definitely Canada. So you have a lot of them. UK is also very involved in this. So top down concepts are definitely a trend. And that means that, okay, they will say, okay, then we need to act now. We need to have either we have our own stuff, we do our own stuff, and that's fine. Or we have someone that comes in, a company that is already doing this and can help us. That's the two concepts. With regards to chronic, this is acute. This is what I'm telling you. It's like now it's all about making sure that you have a system that can do the first part great and that you can use it for that. Okay, and that it's proven to be used and that everything is working fine. Now the next step is of course to nudge on this chronic part, okay, of the preclinical trial. And that is more difficult because then you will need to prove that your models are actually being alive for that amount of time. One. Secondly, if now you're going biology, like literally just not in silico, but biology. And then you have to also prove, of course, that your model is either equal or better in predicting the chronic concept of which is produced now for chronic in preclinical trials is produced by rodents or other animals and so on and so forth. So this is the reality right now. So that part is going to be hard, not absolutely not impossible, but hard. And it will need people from the regulatory aspect to start working more into understanding how this can be translated. And the reason for that is that it would be, in my opinion, not so good, not to say it in a very diplomatic way, not so good to actually, I would say, put in parallel and in this case, look in look if now the results from a rat is the same as the result from this human model, because it doesn't make sense at all. I would rather that we look at this human model, biological model, and look at data coming out for the same substance that this model has been tested in with but for what is happening on that in clinical trials. That would be more, in my opinion, more accurate, because it will go from saying, okay, we have this model that apparently is doing something chronic, we can keep it for a long time, we have tested a lot of things, and that data, we see, okay, can we draw parallels in some way, parallels for some entities, endpoints, or whatever, to what we see in the clinical trial. Mm? That is the interesting part. Not to say we have this thing, we are doing the results, and then we compare those results to another species. That doesn't make sense whatsoever. So this is, I think, the conversation that will need to happen, because we need to be prepared to not make mistakes, other mistakes, or make stupid things, so to speak, just because we want to rush things. We need to be careful with that. So at the end, we can make this, as I tell you, kind of shorten the lifespan, make sure that people get access to even more drugs, and not just that, but actually drugs that suits them. Yes, that suits them. I mean, if you are a person, I mean, I'm just telling you like this. Most of the drugs that we have out there now have been mainly tested for years. I mean, the normal, the ones that we all know about, kind of, okay, they have been mainly tested on males. So that means that the other part of the population has already a disadvantage on this. Mm? Uh, and that comes, of course, with things that we know the disadvantage. We know that the side effects for women are different than for men. We have known this for a long time. Mm? So this is why it's important that we, in this case, are able to model and to do this already in the lab. So when it comes to the clinic, we already have data that we know is in parallel to something that can happen in clinic, because it's the same animal, it's the same species, it's the same. It's we have been modeling women in certain ages or whatever here, and then we come to the clinical trial and say, yeah, so we're going to then acquire a cohort with a similar cohort that in this case matches that. Mm? And then you go ahead and do that. And hopefully, and I'm hoping that hopefully this would then be so much more effective. So then we, at the end of the day, have a drug that actually is really suited for that cohort. Yeah.
So then to go back to CompreVie and your service and product
Mhm.
offering, you focused on skin as the organ of choice, which sounds like it can relate outside of just pharma and biopharma, right?
Yeah.
Maybe cosmetics and other applications. Um, do you offer a service where someone gives you a specific range of phenotypes, like you mentioned, female, male, age group,
Mhm.
specific disorder, that then you can provide that service? Essentially, what I'm asking is, can you provide a little more detail on the product itself that you're offering? Is it just the organ and the chip? And then is there any software that you'd mentioned as well that is, the data collection component
Mhm.
for this product?
So we have decided, as you can imagine, this is like making a small. It's making a living thing in the lab. That means that you need to nurture. You need to know what you're doing. So therefore, we decided with Iphigenia that this is not going to be something that we can sell as something that someone else can do something in their lab with. So it's not going to be a product which can be sold and then someone uses that in the lab. It's going to be a service concept. So it means that we are in contact with the the entity, whoever that entity is. Could be cosmetics, it could be pharma, it could be biotech, and so on. And they will come with a substance. They could come also with, I don't know, a patch that they would like to try out. It could be a cream, whatever situation, whatever concept. Um, and then we would then, in this concept, be able then to say, okay, they are mostly interested in this case, not in in so much in the heterogeneity of a cohort. They are more interested in knowing that this this substance or this scaffold, this patch or whatever it is that they have, how long it has an effect on this tissue. Okay, that's a conversation you have then with that customer or that entity. And of course that can change later. You do an experiment and then they realize that, yeah, but now we would like to do other stuff, and then we can do that too. So it depends on the conversation. So we're going to be a very one-on-one conversation concept. It's not that's our aim, kind of the gold standard that we want to have. Now we will, though, have, like, I would say, off the drawer concept as well, meaning that someone can come in and say, the only thing that I want to know if this substance or this cream or this gel that I'm producing is biocompatible or not. That's the only thing that I'm doing, and therefore we know from regulatory perspectives these are the tests that need to be done for making sure that if this is biocompatible or not. And then we just take out this off the shelf concept, we put a normal scenario of a tissue, and then they get the results. Okay, so that could be just an off the shelf thing that can also be serviced. It's not a problem. But the one that we would like to kind of evolve more into is the other one that I told you about. So be able to really cater to these things, because that's the one that is going to give you, of course, this predictive concept more. And that's the one that is going to be able to give you also the chance to have something to go or hopefully circumvent the in vivo part of preclinical trials. So being able to do so. So that's why we went for the service. Now, do we think that we can do the service ourselves, or if it can be licensed, for example, the concept? Yes, of course. If it is a big pharma company and they have already, I don't know, a lot of people working doing modeling, and they would like to test this out in their labs, of course we can collaborate, and they can get a license to use this technology with them. So it's not we don't see that as a no-go. We just don't want, I would say, um, uh, people to feel that it is too complicated for them to use it in the lab, and then they just give up. And this is what has happened. Just as a side note here, one of the challenges for organ on chips, why? Because organ on chips has been around for a long time now. I mean, it's not something new. But one of the things that has happened and why it has gone, not just because of the regulatory things that have taken a little bit longer, but also because people are having a little bit of a trouble understanding how to use it, um, and how to go from this really easy petri dish concept to this other thing. And can I really trust it? Will it give me results that I want? All of these questions, okay, which are understandable, because biologists and chemists, we are all very kind of, if something works, we're going to stick to that for a while, because it's otherwise going to be a lot of variables that we need to still think about. So I completely get it. So that means that with that said, with that mindset, both of you and I said, you know what, doesn't make sense to put that headache, that enormous pressure on a customer. Doesn't make sense. But if now the customer comes and says, guys, we have a team that has been working with organ on chip concepts that is really good on this. We have already done some organoids, and we will just want to incorporate your system in ours, and could we license it? No problem whatsoever. I mean, that's what we want. I mean, in a way, I would love everyone to be able to use chronic testing and so on and so forth. So cool, super cool if that would happen. Mm?
So we're using this as a service for those who may need some support, but then also the ability to license it
Yeah,
to teams who maybe have the expertise in
exactly.
house.
Exactly.
Okay.
That's,
That's the best way to do it, right? Try to
yeah,
be flexible with your
be
offering.
flexible with the options that you have, especially when you're so young, and especially when you have something like this, which is extremely deep tech and it's extremely, I would say, time consuming to set up if you're just setting it up. Okay. It is not like going, okay, I'm going to make dinner and that takes you 30 minutes. That's not it. It's not just following a recipe. It's more than that. You need to have a very good team behind it. You need to be able to, which I'm not saying doesn't exist anywhere else but in CompreVie. I think we have so much talent out there that I think that you could do this kind of concepts anywhere in the world if the facilities and if the money exists. But to make things, as I tell you, to make it go from being something that people just hear about and think is cool, organ on chips, oh, super, super nice and very cutting edge, to go from there to have the same thing that we have now with the phone, with an iPhone, with a smartphone, that everyone doesn't really think about the fact that you have a smartphone. You just have it. This is what I want. This is what we want. We want it to be something that is completely normal to use in a preclinical setting. That's kind of a, maybe not a, hopefully a gold standard, but that is there. It doesn't need to be even only a CompreVie concept. As I told you before, this market, this uh, space of research, this space of development, commercial development, is buzzing. It has a lot of different companies coming out with really interesting and innovative stuff. So the more this grows, the better. But we need to also be able to get people to understand that they can use it. They don't need to be afraid of it. It can be used and do something with. Hmm?
Speaking a little bit more to the fact that this industry is growing and CompreVie is newer to the market,
Mhm.
what is your strategy currently to bring yourself awareness,
Yeah.
get more beta testers,
Mhm.
partnerships, etc.?
So at the moment, our strategy is to have as many pilot testers as we possibly can with the people that are on board. We are a very small team at the moment, and hopefully we
Okay.
can grow this year a little bit. But still, uh, and with that said, the pilot testing we know already has, I would say, a ripple effect.
Okay.
Usually, because people will say, okay, no, it worked super, super nicely. Why don't, I mean, then this other company comes in or this other one comes in and we can start making a small, I would say, network of
Okay.
those that would like to try it. In my opinion, or what we want to have is that our first kind of launch of the service platform with certain, I would say, it will not be the full Monty, so to speak. It will not be like this product, the service platform with all the gadgets that I told you about yet, but it will be off the shelf concepts that I talked to you about. And of
Okay.
course, also limited amount of those that are one-on-ones. That's what we want to launch with the skin model concept. Having maybe hopefully some predictive concepts there as well already, and that will be the scenario, and that we would like to launch, I don't know, 2029. That's not that's
Oh,
that would be
okay.
like where we want to launch it, where we think also that we are able to do so. So we
And
are
this
at
would
the
be the
moment,
first commercial product.
then it will be this service
Gotcha.
and so on and so forth that we launch then, because at the moment we are just a pilot testing concept, so we can then uh, I mean, we can try things out, we can make sure that everything is set and so on and so forth, and then 2029 we want to be able to say, guys, now you can come in and we can take these orders and we can do this. It's like a pre-war, it's a pre uh, the pre-opening of a restaurant, so to speak. That's where we are now.
Okay.
No. And then you
Okay.
go for the opening, which will come then. Is if we everything goes as we want it to go, 2029. That's where we want to achieve it. Mm?
That's great.
Mm?
That's great. So going into a little bit more information on CompreVie,
Mm?
you've recently won an award. I believe
Mm?
it was the Rudolph Salinger Fond's SMB Award.
Yeah.
Do you mind sharing a little bit about that
Mhm.
and what does that mean?
So it means that it's an award for science and business. So it is an award that is quite well known in the sphere of this region, so to speak. It is, we got what is called the public vote award. And that means that people voted for us, for our company, and what we are aiming to do. So for us, that was super nice because we, of course, that's actually even sometimes means more than getting a chunk of money in this case. And for us in the pilot testing concept, it's even better because then you have uh, this other platform that is giving you them the opportunity to say, oh, they exist, and so on and so forth. So one of the things that we got from there was, of course, to be interviewed for a magazine that is very popular for business here in Austria and also in Germany and so on and so forth. So that was really nice because we've got to, of course, also explain what we do and what our achievements are, where we come from, and what we want to do. So that was good. We are also hoping, of course, I mean, we know that this is an award, but we are hoping that we are able to also attract more people to be aware of the situation with such an award. We have been also nominated for another one, and we were part um, of that award ceremony. We didn't get the award though for that one, and that was Global Startup Award. That was also quite interesting. And there you, of course, have a broader sphere. It's more about uh, what is new in the region. So in this case, in this part of Europe. So that was quite nice as well to be part of that. So we have tried. Corinne, we what we try to do is to make sure that we are as visible as we can be with the budget limitations that we have. Of course, you can go completely crazy, and if you have the money for it. But being a young startup and being also extremely involved still in R&D, and making sure that we spend the money wisely on that, and being able to maybe travel to one or two conferences, things like that, we feel is more important than having an agency that takes care of all of these things and puts our name here and there, or pay someone for giving us an award, or all the constellations that exist there. That for us at the moment is not, I mean, we don't have the budget for that, so we try our best to do what we can, and therefore we are very, I would say, we try to be as active as we possibly can on LinkedIn. That's our platform of choice. And you can always literally reach us via that. I have also, again, the privilege of working with Iphigenia, who is extremely artistic and very good in everything that has to do with, I would say, the visual part of the company and also marketing. So it means that I can rely on her on a lot of things, because me, I'm all about ideas, and kind of my head sometimes is a fruit salad. So it's good to have someone that can take over those things, because I just don't have the time sometimes to do that too. So it's really nice. And she's very gifted in that. Mm. So this is how we have done it so far. Of course, we hope that with the years, until we are going to launch this service, we are able to increase that budget so we can be part of more things and go to even more meetings and even have events. So this would be something that we would dream of having, actually, that we, if Ifigene and I, can put up an event here in Vienna for people that are interested in knowing more about how to use these systems and so on. That would be super, because again, it's all about embracing, making sure that people start understanding this embracing of the complexity of ourselves as humans, and how we can model that in real life, and how we can model that in the lab. That's that's uh, and I think that that comes with education. You need to educate people on that, not to be afraid of it, but actually just embrace it.
We'd like to thank our guest, Elizabeth Pavez Lorie, CEO of CompreVie, for joining us on the Biotech Stack. For more information on Elizabeth or CompreVie, please check the links in our show notes below. Thanks for listening, and we'll see you next time.